I want to start with 4 times when I found myself being fooled, and I want to see whether you were fooled also as a lead-in to where I'm going. So I'm going to start out with 4 examples of where I think all of us in some ways were being naive. First has to do with expression profiles. I had a chance to grow up right at the right time when we looked at the power of like hanging all these microphones in a cell and asking what was going on. It seemed like spectacular. Well, as Lee Hartwell and I said, we can look at the what without asking the why. And that's a very important concept, to look at the what without knowing what the why is. And here's an example of a paper you may know. Tim Hughes did it with us. And what we did was we used heterozygous and homozygous knockouts across the entire yeast genome. And we built up a compendium of 300 important one of those. And then we used that as a way to go in and query unknowns, unknown conditions, unknown compounds. And we found that it was remarkably informative. And this actually has been a concept that has grown in terms of the power of it. But I want you to step back and see whether you felt the same way I did, which is you can go into any tumor type. You can go away from cancer, but let's stay in cancer. You can go into any tumor type, and I can find you 6 different papers that all go, I've just built a beautiful classifier for X, okay? And they're all different, okay? And we should be asking ourselves not who's a fool or who's lying, who's doing plagiarism. That's not what's going on. Right? It's that if you look at a situation, what you realize is what you think is the same situation, what you think is looking at the same problem is not. The second example of being naive comes from looking at DNA alterations. As we shifted from having occasional links between cancer and chromosomal aberrations to having detailed component lists, there has been out there a concept that if we could just get the sequence done on every, this is interesting. Let's see what I do. Did I touch that for a second? I guess so. If I could get, on every patient and every part of their tumor over time, their whole genome sequence done miraculously, that somehow we'd know how to treat people. So the point I'm making is there is out there a concept that altered component lists at a DNA level is all you need to have, right? You can make sense out of it. You'll know this patient's going to have a RAF-altered melanoma. Give them this, give them that. And I think you have seen that actually is also an illusion. And it's a wake-up call in terms of what really goes on. This is a beautiful paper, came out 3 or 4 months ago by a friend of mine who, like Eric Schott, and if you ever really want to talk to Eric Schott, talk to him about speed, talk to him about racing. Here's Frank McCormick, another person who's like infatuated, not just with science, but with speed. And this is a paper they put out, and why I've put it up is this. This is a RAS signaling diagram. Starts, I think, with the same point that you began. And yet, what this paper showed, because this is important, is that if I make a mutation in any one of those genes that are noted, so here's a KRAS and an NRAS, that actually the function of the other changes. Okay? So it's not just I'm going to look at the mutation here or there, and then I have knowledge. This showed that actually you modify, it's like having a giant tumbler, and you switch one dial over here, it actually changes everything else. Okay? And that's the complexity that we have within the cell. Similarly, those of us who are thinking, work at a DNA, RNA, even a protein level, let's go real, let's get to functional data, who began to work on functional screens and said, we'll go to where we really make sense of it by going to functional screens, and worked on synthetic lethal screens. Same thing happened. Here's a paper from when I was working with Alan Olaf at the Fred Hutch, showing how you could find synthetic lethal mutations. Actually, this was building the alterations that were in human genes into yeast and running those screens. But here again is that wake-up call. Are we being realistic about what goes on? I could pick 8 examples. There are 8 different papers out there in the literature about what happens when you run a synthetic lethal screen on RAS. I picked 3 because these 3 people I work with, I like them, and I guarantee, no, I work with all of them, I know them well, but all of them put out a paper. You go in there and look at it, and it has a different wiring diagram. Okay, it says these are the synthetic lethal genes with RAS. This is the synthetic lethal genes with RAS. This is the synthetic lethal genes with RAS, and they're all different. Okay? So this concept, whether it's at a DNA or an RNA level, at a protein level, or a functional level, that there's some big truth out there, matches if you talk to any clinician with what goes on. So if you go to those who are treating patients and you take these 5 drugs that we helped develop at Merck, so I worked on all 5 of those drugs, and you take those into patients, you know what I'm going to say, which is the phase 2 trial doesn't look like the phase 3 trial. And it's not that someone messed up. Sometimes people mess up. It's not that they messed up. It's just that the heterogeneity that sits there, that's real in context, and the importance of context is very high. And so to a diagram, I think we'll each have one of these diagrams. The point I want to make, throwing this one up there, is remember that evolution has hardwired us so that anything you do at any place, any change you put in, there are redundant feedback loops that you cannot get to by thinking I'm just going to do one simple peel off the onion and now I know what's going on. And I bring this up because I think it fundamentally puts a skewer through the way we do research. Okay? So we do research and the NIH does funding as if what you really need to do is hypothesis-driven research. How many people have heard the phrase hypothesis-driven research? I'm going to see hands, okay? How many people know not to put in a grant that is not, right? That you have to put in a hypothesis-driven grant? I know. I've tried otherwise, okay? But what happens if that's not the right way to query, okay? What happens if, how often are we hurt by going from the particular to the general? What most people do is they go, I'm going to work on apoptosis, or I'm going to work on stem cells. And they go in and they find a particular little finding, and they build it up, and they see if they can get the editor to buy that this is going to be, the answer to cancer, right? And then if it is, it goes into one named journal, okay? And you get your first Lath author, you get your high H factor, okay? And if you can't quite make the story, but most people are incorrectly trying to generalize from the particular. And one of the things that's on the bottom that I worry about is often when you go to publish your version of it and someone has published something to the side, they go, oh, well, I know that's wrong because someone else just found it. It was different. So I actually think we hurt each other and we hurt our understanding of what's going on in doing that. There's a beautiful paper by Trey Eidegger, who I've written with, I know well, that I'd like you to go back and look at because actually in this paper, he shows that you can build an ontology, okay, a hierarchy of relationships of what does what to the cell, not going to look at the literature, not going to, I did this experiment, but literally going and building it up from the facts within an experiment. You can build an ontology, right, starting from scratch. And the paper that he's working on, that we're working on together, is to show that actually ontologies are context specific, okay? This will be very important because right now people are saying, well, if I just know the roadmap, then I can match everything on to the roadmap. Well, what happens if the roadmap is context specific? And I'll bet it is. Okay? So if that's the case, then let's plot ourselves on how far we are to being modern scientists. And I think what you'll see is where the chemists were in the 1700s pre the periodic table. And we have to stop kidding ourselves. We're alchemists, okay? We're no better than alchemists. We pour a little this and this, we get this together, we file it, and 200, 100 years from now, people are going to look back and go, oh, those scientists, they all thought they could take this to that and make that into a metal, make that into that. And they'd look at the Hanahan, Weinberg, Hanahan, what I call horoscope. This is this is that. And they'd line it up on the horoscope and they say, this makes sense. No, I'm really quite serious. And having trained with Bob Weinberg, I don't think he'd mind that analogy. We don't know what we're doing, okay? And the patients are dying because we can't figure out the rules to what we're doing. One of the most important evidence that we're shifting out of this is a very important paper that came out from the Institute of Medicine a year ago, December, called the Precision Medicine Report. And basically what it said is, what we have to really start doing is start collecting and building a knowledge network. Not saying this is right, this is better, this is wrong. There is good and bad data, but I'm talking about not putting judgments in and putting, whether it's the exposome or the epigenome or the microbiome or the genome, and begin to try to make links between those and to build an information commons if we're going to have a new taxonomic classification. Right now, we fund people and we do our work in ways where we start with a biologic system, we generate our data, we go and do our analysis, we publish our paper, and there's an automatic linkage between the data generators, analyzers, and validators. And this actually is the way physicists worked in the 30s. They don't now. But it is almost a feature of doing hypothesis-driven research. And this is important. I don't think that hypothesis-driven research is a bad idea. It's just that we have these cool tools and important ways to query that should complement that. And my point is that hypothesis-driven analysis, where you have a lab of 12 people, you have 4 postdocs, you have 2 graduate students, I'm working on how this binds to that, right? You can do in that way, okay? But some of these problems actually need to be looked at a broader, more sort of large scale, the same way as the physicists went off their lab bench when they stopped doing those little experiments on looking at alpha particles and realized they could use large colliders. Data-driven data analysis is such that actually it's very valuable to have people who did not generate the data do the analysis, because there's so much data to look at that it's actually very valuable to have a loop in there where the data-driven data analysis does not have an automatic loop. In fact, there's an uncoupling between those that generate, analyze, and validate. They all need to get together. And in order to make that effective, I'd like to go to alternate to Moore's law. I think everyone knows Moore's law about efficiency. But there's another law Metcalfe wrote, which had to do with how fax machines and how the internet work. And it has to do with what happens when you can have compatibly communicating devices working together. And it's a very simple law. It just says the power grows as the square of their numbers. And you have a lot. If there are only 3 fax machines in the world, it doesn't work very well. If everyone has a fax machine, everyone's on the internet. So the thing that I've been wondering about is, why doesn't that apply to biomedical research? And the reason that it doesn't apply is we are stalled because our communication devices today are articles. And we don't really want to tell someone what we're doing until we get it accepted. And so there is a gap between when you learn something and when you're ready to talk about it because you don't get credit for it. And so this slows enormously the way research is being done. And if you're going to do hypothesis-driven science, fine. But if you're doing data-driven science, you need to be able to have that communication. And this last week in Nature Biotechnology, there's an article that I wrote along these lines that just came out. Next thing I want to go to, if you're building a center and you're trying to figure out, do I need a program, do I need a department, et cetera, is there is a discontinuity between the state of the technology and the state of institutions. So I did some of my training at Harvard. This is a classic picture of what a university looks like. Today, the data that's coming out and the data you want to get your hands on is this genomic information that can be done anywhere, maybe cheaper in China. There is a willingness of patients to be participants and give data and give insights that don't require them to come to a clinic within a hospital. There is this ability to work on, to build up networks. And very importantly, as I'm going to get to later, there are ways to have projects where you don't have PhDs doing that work. So I hope you all know this project, Fold It in Eterna. There are enough blank faces that I'll take 1 minute to do this. So go look up Eterna. And Eterna is a project that came out of Berkeley. And this group wanted to know, could I take the most complex puzzles in protein folding and could I actually gamify them? Could I put them out so that instead of the postdoc at Berkeley being the person who has to solve that crystal structure of folding, could I turn that into a game? Could I put that out there into the world? And you may know that in this effort, it took 3 years to put together, a guy at Carnegie Mellon who's now running efforts at Google, so he's moved to Google, Adrian Truel, showed that he could get the whole world to engage, whether they had any degree in math, whether they had any degree in biology. And the woman who won this was a housewife in Surrey. She'd put her kids to bed at night, and then she'd go in and she'd play, I'm being protein biophysicist. She beat everyone else out because she had passion, okay? When you have passion, degrees don't matter, okay? You come in, you energize, you couple to those, and when asked what did she want as a prize, she didn't say, I want money. She said, I want scientists to figure out whether I'm right. Okay? That's the energy that's out there. And the question is, how do you use that in order to get to information instead of our classical approaches, funding one center, funding to have X number of faculty doing this or that. So 4 years ago, Eric and I co-founded Sage BioNetworks because we got interested in how you could get ready for this world that we think is going to fundamentally change. And how can you build a way where there's open, collaborative ways where teams of teams that are working not in the same building and go beyond the current guilds of experts. So I've been running a nonprofit in Seattle that has 30 people for about 4 years. And you can get on our website and go through and see what we're doing. Half of them are software engineers. I'll show you what they do. Half of them are researchers. And that group, on an average basis, is putting out 2 publications a month. And we don't generate data. We don't validate data. We just take data that's out there, work with it, make insights. I could go through this example, which is how you build a RAS model prediction for sensitivity to EGFR inhibitor. But for teaching purposes, I want to go to a non-cancer example. In fact, it's one that some people in Eric Schott's group, and maybe Eric, you were on that paper also? Yeah, Eric and Bin worked with Chris Gaitari on this. But I want to show you what I mean. So I'm going to drop down and do some science before I move to the side. So what we wanted to know in Alzheimer's, starting with hundreds of brains that had been analyzed for expression, copy number variation, was could you build models off of this type of data? So the first step was simply to go all the way through the genome and look at expression and see what gene acted like any other gene the most. So it's a very simple step. It's very easy mathematically to go and build modules of co-expressed genes. Incidentally, this just came out in Cell this week. Okay, so you can go on to Cell and look at this. And so all you do is you build co-expression calculated for disease versus the healthy disease. So we took the Alzheimer's brains, took the normal brains, looked at co-expression differences. The second is we then prioritize those by looking at the synchrony between the clinical measures or tendency to reconfigure them. So we did a rank order of these clustered co-expressed genes and said, well, these are ones that almost all the time, all the way through every parameter, and you said that may be more important than others. And then we went up and took genetic information, in fact, eSNP information, and looked to see if we incorporated that genetic information, could we begin to find directed relationships between the genes? You know, as was pointed out, if you just have 2 facts, you can only get to associations. But when you get triple layers, you can begin to look for more causal relationships. And when you do this, you can go in then on a particular question, looking at microglial activation in Alzheimer's. You can do the eigengenes of the modules. You can look at particular probes, and you can begin to ask what are the controlling genes, the genes which are most likely to drive the effects in other genes. And what comes out of this, which blew our mind, was that so this is Alzheimer's. Sitting in the middle of Alzheimer's, the core drivers were 5 components: complement, the Fc receptor, MHC, chemokine, and the toll-like receptors. shown in these colors. And so what was going on in these brains with Alzheimer's that was driving it was actually inflammatory reaction that was really the difference between what was going on in the normal and the Alzheimer's. For those who study inflammation, they go, yeah, another one, okay? But it's pretty remarkable because it didn't come up with, here is the A beta, here is tau, here is the neurotangle, okay? What was driving what was the crud that you pick up when you look at the pathology, yeah, is that. But what was driving it was actually this tightly interconnected web. And you can then reflect that information that you have there and look at something that is more classical for us to look at. And so in red now are all the components that we picked up that were actually important. And the one that was one of the most important was a receptor, TYRO-BP, or sometimes called FCER1G. And that particular protein sitting there next to TREM2 was able to be tested by doing RNAi experiments where you do full-length truncated versions, looked at vector controls, look at the microglia, and could show that actually validate that in fact what you thought was driving the circuitry was. And what we now have been doing is to take those gene regulatory modules, begin to try to overlay those with the diffusion spectrum imaging, looking at the microcircuitry. And Chris loves this stuff. If you're interested in any of this, go and look. But what I want to do is just take you take a bunch of data, you go through, look at the co-expression, you look at the priorities, you layer in these steps. The point is, this is not black magic, okay? They're steps that you take, put it together, you can make sense out of it. When we began, 2009-10, we were wrong. We thought that you could put a lot of data out there and begin to get people to share and work with each other if you made cool data available. And what we found was they would sort of look at that and they go, but I really was working on this and I might be able to borrow that, but I'm not that interested. So one of the things that I think the Allen Brain Institute, other places are finding is spectacular data made accessible, even curated and made usable is not very helpful to try to get people to work together and work as teams. We then did a project called the Federation, which was after Star Trek. We thought we all have a common enemy, and we all can't get there on our own. So the Federation initially consisted of Eric, myself, Trey Eidegger, Andrea Califano, unknowns, no, I'm just kidding. And then Gary Noland and Mark Vidal joined. And this group of people said, let's let all of the graduate students and the postdocs have access to all of the data that each of us have, all of the tools, everything that's in one of those labs. Let's just show the world that if you work by yourself, you're not going to get there. But if you join and you make rules for sharing within a group, you could form a federation. And our goal was we would do that, and then everyone else would go, well, wait a second, why can't I be? Right? Why is this exclusive club? Why can't I be in? And then we go, well, here are the rules. Right? And the second half worked, right? So we have all these people that want to join the federation. Can we join the federation? But when we started this effort, we expected the postdocs and the graduate students to go, this is fantastic. I get all these people to work with. And what they thought was, oh my God, now Gary Noland's postdoc is going to take my work. So they felt this was threatening, right? We thought it was a fabulous idea. But they said, I have a pretty good project, but I want to keep my own data. I want to, and we were discouraged. So this didn't work, okay? It was beautiful for us. We thought, get rid of the silverbacks and they'll all play together and have a good time. But they were, oh my God, right now I have to be careful. So what we began thinking about was how do you change the incentive structure by which, to get people to share. And we realized that it's not like minor, it's like major. The real incentives are first and last authorships in prominent journals. I mean, it's just, first of all, science has become a business, okay? I need to support my family, I make my employment from my work. I have to be a PI, I have to get tenure, okay? And what sits behind that is, gone are the days when you sort of did it on the side in a way where the patient was center. Those days are gone. And so we thought we'd better come up with a solution for this problem. Otherwise, we're going to keep running into people who want to do it, but don't. So journal articles have been around since 1600s. It's like a little over 400 years ago that the first journal article, it used to be that scientists would write to each other. They didn't have any phones, but they'd write to each other. I found this, I found that. And the Royal Academy, and this is the first article, right, one of the first articles published in the Royal Academy, was a transition. And what we realized was maybe in this digital age, there are better ways to communicate. That maybe there are better ways to talk to each other. And we basically borrowed directly from a project that the software engineers started about 4 or 5 years ago. So if you look online at GitHub, GitHub, it's only 4 or 5 years old, but it has grown to be a place where now it's 5 million different repositories are, and 2 million software engineers are working. And here, every code changes version, every issue tracked. Projects, when they start them, you can see they can evolve. This is important. It's both open and closed. So Microsoft runs its programs in a closed part of GitHub. There are other parts where the entire thing is open. So if you had a department, if you had the whole UAB and you wanted to just share it among some cells first, or you had a consortium, you can tie that down like that. But it is a place where you can say, I want it to be this open, I want these people to be working on it, and the most important thing is they get credit. Why is this popular is that it has gotten to the point now where when you apply at Google or Amazon for a job, they don't ask for your CV. They don't care what you have published. They don't care at all. You know why? They can look exactly at what you did, okay? They don't want to know what the head of the lab did, okay? They want to know what you did, okay? And this, so it's paid for by the, basically the HR departments. Every company wants to go in and look at who's done what. So they have transitioned from papers being, which the way you keep track of it, to going, just show me what the person does, right? What are they good at? And so we thought, let's build a GitHub for biomedical scientists. Let's have it so that you could go on, and when someone is developing a model, or they've curated data, or they've done something, you could get credit for it. The analysis history, the conditions were there, where you would then be potentially willing to share. And so we've built this system. Many of the people who are within our group worked at Google or Amazon. And you can run it on Bioconductor R, or RStudio, or Python, but it's all recorded in Synapse. So Synapse, if you remember one thing from today, those guys were working on a geek sandbox, okay? This is a geek sandbox, okay, is what it is. And it is for private and public workspace that allows you to aggregate, describe, share your research. But really what it is it's a collection of living research projects that actually now can follow Metcalfe's law. That's the reason for putting this together. So I didn't trust the time in terms I wanted to go live. So I'm going to do some screenshots that it would be if you were going on. So if you can go on, one of the larger projects we have is we took all the data in TCGA, dropped it into Synapse. And the groups that are working on it actually are now all working on Synapse, so you can click on that, you can go in, you can look at the core projects, any tumor type, and here it's in a way where it's curated, it's annotated, so you can get to it other than an Excel sheet. You can go in and take a particular thing that you want to look at, like this is these music analyses. You can click on that, you can go in and ask who's doing what. Here comes the important point. You can go in and see that Syriac was doing something that Kyle Elrott had gotten, and they had gotten that from that. So now, when someone has an insight, right? Not data, but now I'm talking about an algorithm. I'm talking about building something that everyone can use. You can go and you can see that, and everyone else who's ever used it, they get credit for it. So your goal now is to get 1,000 people working on what you have done instead of being frightened that someone borrowed it. Okay? You want people free writing on your stuff because you get all this credit as people go off and do it. So you invert it. So you want people to use it. You want people to take it. And your credit is driven. This is provenance. Provenance is at the heart of Synapse. And I think that's what we found allows people to begin to share. So now you can go in and you can look at the attractor metagene data in pan-cancer, and you can get different people, before they've ever published it, different people's tools on how they've gone about looking at the data in real time, because they get credit for it. We could go and we could take the CCLE. There's this big data sets that were sitting at the Broad, they were sitting at the Sanger, and we put them into Synapse, and now you can go in and take in and out all of the sort of modules that made the work. So you can go in and you can put plus or minus expression data and compare two people's data sets. So instead of looking at papers, you can go in and look at the actual work that was done, what algorithm, and you can say, that was important, this wasn't. This is work of INSOX. Also for a project I'm about to talk about later on looking at challenges, we found that it was very helpful to be able to have leaderboards where people could see who was beating each other. And what we found was if you want to get people to share, have them out-compete others. Best way to get people to share is to have them compete, right? Because they hate being number 2 or 3 or 5. So put it up on a leaderboard, then you know where you are, and you go, I hate those people at Harvard beating us. You know, we're going to show them that we're better than they are, and they stay up all night, and they connect with another. So we found leaderboards and visibility are very helpful in getting people to work together. You can click in, you can see the code, you can work with the code, but if you modified it, it tracks, you did that. You can go back, and it basically, in parallel, goes all the way back when you do that tracking. I think you get the point of that. You can go in on a figure, you can click on the figure. When you click on the figure and you get to a point, you go and you can see where it all came from. So you can go in automatically and have this provenance work out for where you are. So I think in June there's going to be a big series of articles that are coming out in Nature, I think, and all those will be so that you could host them on Synapse and you'll be able to see them there. Now, you know who this is, Gordon. One of the reasons I'm really happy is it's okay to work with genes, but I got my Ph.D. in protein chemistry. And proteins are important. They're actually getting almost usable, as you'll hear later. And the Cancer Protein Atlas, Gordon was kind enough to give me a clue was going to become available. And so now we're working to make sure that the proteomic data that he's like brilliantly put together is able to be hosted up onto Synapse, because the same thing could go there. Now I'm going to go to the next level of functionality. I have 1.5 to go. One of the things that people have found in the digital age is that if you have a lot of data, you can put this information, whether you're doing the Netflix challenge, whether you're doing Next Prize, whether you're working on an incentive, out, and you don't have to pay people to build those answers. You can get it by giving them a prize. One of the advantages of that is you pay for product. You don't pay for someone who isn't able to do it, and you don't pay to one person. And so we have been interested in figuring out how you could take large amounts of data, get to predictions, have unbiased evaluations, avoid the overfitting that almost every scientist does, whether they try to do it or not. Almost every scientist overfits. So can you make it so to get around some of these issues on overfitting and measure things so that you, when you're going to talk about whether you've done better than another, it's not, I have more citations, but actually, can I outdo, can my algorithm outdo your algorithm? So transparency, reproducibility. So we started just about a year ago because some other friends of ours had put together records, and now I want you to think about what you have at UAB. So they had 2,000 patients who had clinical records and they had genetic records. And the reason I was interested in this is 10 years ago, we had the Nature and New England Journal articles that were on the first example of finding prognosis for breast cancer that turned into MammaPrint, turned into Oncotype DX. We did this with a group in the Netherlands, and it turned into a $4,000 test that patients have to pay. And I know it only costs $50, and I'm irritated by the fact that it's not moved, it hasn't changed, it's become gospel, and it's not actually dynamic, it costs too much. So we thought, let's blow that up. Let's see if we can get the crowd to build a better classifier and have it in a way where it's open, and then someone could build an assay, and you could use it in the third world. You could use it a lot cheaper than the guys at Genomic Health sell it. So we had the data set there, and we realized we had the training data. We could make it so that you could submit your models to Synapse. All those model predictions could be evaluated, and then you could get a journal. So what we did was we lined up with the editors at Science that the winner of the paper would automatically get a paper. Forget peer review. Just blow up peer review. No, seriously, why have peer review if you've beaten everyone else? Okay, and they agreed to it. Okay? Bunch of conditions that had to be in it, but we made it so the winner of the challenge got a paper in Science Translational Medicine. Then we got Google to say they'll give us a 1,000-node cluster and make it available so anyone anywhere in the world didn't have to worry, I don't have a big enough cluster. So whether you're in a high school or whether you're in another country, as you'll see, anyone in the world had the same access to available space to carry it out. And we divided it into 3 phases. The first phase, we took 1,000 of the samples. There was unlimited model submission. We had 500 that were left out to train on. We went to a second phase where then they were restricted to 5 models per team. And then in the end, we had an entirely different set of patients. I wanted to have the winners have to actually perform and to characterize a set of data that no one had ever done. In fact, we were generating the data during the challenge. It was a little risky, but it was women in Norway that we generated the data on so that we know that if you won, it wasn't because you'd overfit the data. Entirely different population. And this was all done because the group at IBM called DREAM had spent 6 years every year going and doing 3 or 4 of these challenges. The second thing I want you to remember is what DREAM for us means is a dialogue in reverse engineering to assess models. The D is the important part. We wanted to build a community that was open and learning from each other. It wasn't just could we find the best answer. You can do that with incentive. We wanted to build a community where you were rewarded for working with each other. And so what we found was, in the end, over 35 countries participated. We had hundreds of groups. We had thousands of models. And within 6.5 weeks, this group beat those of us who were, quote, in the federation. So they beat the UCSD group. They beat the group at Stanford. They beat the group at the Broad. So it took the crowd about 6.5 weeks to beat the groups that get most of the money, as if they're the anointed ones, top down, right, and said, let's just give it to the whole crowd. And more importantly, this is the third important thing I want to go over, the person who won it, when I read the paper that he came, it was a very awkward moment, I would have rejected his paper. So if I had read his paper, I would have rejected it as an editor. So you might go, how did this happen? Right? Where's my bias? Where's my prejudice? Okay? The paper said, I've solved the world, da-da-da-da-da, didn't really give what it was done. The method that he took, I also would have said, shouldn't do, because he didn't do stage or grade. He didn't listen to any of the clinical parameters. He was an electrical engineer who said, just give me every tumor I can get my hands on and the data, and I will build a model that actually will try to look at the core components of what causes tumor cells to be tumor cells. And he was pretty smart. He was the person who solved the MPEG data compaction, and Columbia gets $170 million off of his patent. So he's a smart guy who works outside the area, who no one would pay attention to because he wasn't paying attention to any other things we think, right? So he wasn't one of us, okay? But he blew everybody out. And so that got us the cover in Science Translational Medicine this week. There's a cover story on it. It's in the Wall Street Journal. Sanjay Gupta covered it. But the most important thing is we saw what the crowd was able to do. And so there were scientific conclusions from doing that, which was you could build a model leveraging expression patterns from large-scale genomics. The model performance was generalizable across independent evaluations, getting away from fitting. You could leverage the wisdom of the crowd. But more importantly, why we did it was actually not for the science as much as for how do you build communities and how do you build a template for future challenges. How do you have generalizable software that people can come in and use? And the day that the paper came out, people had been in the background knowing it was going to get published because there's a delay between acceptance and publishing it. And they had gone on and they'd basically improved it. So on the day it was published, you could go on to Synapse, you could see the paper, but more importantly, you could see 3 people who'd run ahead of it and actually had better answers than the one that had been published. So Gustavo Stolovitzky is a brilliant physicist, scientist, who had worked with Andrea Califano for a while. He built this effort called DREAM. In February, as he tells it, we got married. I didn't tell my wife that. But Sage, BioNetworks, and DREAM came together, and that whole IBM group folded into Sage. So now their expertise and everything from IBM has come into Sage because it knew what they were doing. So you might ask, so what are we doing this year? So these are the challenges, and one of these I think is pretty close to what you're doing here. So there's a whole cell parameter estimation challenge. There's one with the National Brain Tumor Society. There's one with NIEHS on toxicogenomics. There's one with the Heritage Provider Network Foundation on breast cancer, and one on RA. The DREAM whole cell parameter estimation challenge is for you bottoms-up parameter equation system biology, classic system biologists. It took into account that actually there's a lot of data out there, but hard to understand it. And really, the question was, in between that data and knowledge, how do you build those models? So this was one where the challenge is about building models to predict cell physiology. And I think this one is in not yeast, but mycoplasma. And I think that's where this one is built. There are a subset of 50 to 100 parameters that are basically sitting at the center of it. The goal of it is to actually come in, use your computational methods to see if you can go ahead and make some predictions on what's going to happen in the experimental data. There's leave-out data, there's training data. So you've got the equations, you've got the data, you've got the leave-out data. Stanford has provided the model. Collaborators are with Stanford and MIT. It's sponsored by IBM. The second one is by NBTS. It's about glioblastoma. I was very happy to learn, Yancy, that you were one of the co-chairs of that. I didn't know that till I came to UAB today. But I had thought it was a good idea to work on it. And this is a beautiful project by Andrea Califano that he and I are co-leading with help from the NCI. And it assumes that actually, as I think all of you know, glioblastoma, it's been extremely hard to find good therapies for that. There are mouse models. More importantly now, there are good models where you can take patients' samples from their brains, from their tumor, and actually grow it in the mice and run the drug sensitivities there and match it to cell lines. So this is being driven by TGen, by Mike Behrens at TGen, by Albert de Jong at MD Anderson. So we're using their models, plus models from around the world that are xenograft type models. And then the Broad Institute has basically made available all of their data on the glioblastoma lines across. So we'll have sort of a combination of the data in vitro, we'll have the data from the patients, and the goal is to see if it's a two-phase project to see who has predictive in silico models and then to use those models in the second phase to see if people can computationally predict combination therapy. We're interested in seeing whether you could take, if you take two drugs by themselves and you do that data, who can predict the combinatorial data? Right? Because it'd be nice to take classical agents, put them with the new drugs, but it's hard to afford to do that. The Heritage Dream Breast Cancer Challenge is one that is looking at sensitivity to mammary cancer cell and therapeutic agents. The challenge is actually to see if it's possible to map the signaling networks in breast cancer cells, and this is one that should be very familiar to Gordon because, yet again, Gordon came through. This is his data. It takes the reverse phase proteomic data on 4 breast cancer cell lines, 88 drugs, 72-hour time points, so that there are 11 time points, 72-hour time courses with 11 time points. And the challenges will be to infer the active pathways in different breast cancer cells, to predict the response of breast cancer signaling networks to therapeutic interventions, and devise innovative strategies to visualize this type of data. It's being put together by MD Anderson and Joe Gray at Oregon Health Sciences, and Laura Heiser at, but just that's how many people it takes to have behind the scenes in order to be able to put on a challenge like that. In rheumatoid arthritis, there's a block of therapies called anti-TNF therapies or TNF therapies. The response is about 50 people percent respond, 50 don't to anti-TNF therapies. And no one has been able to figure that out. The drug companies have worked, institutions have worked. And so Robert Plenge at Harvard has been working with us, and the idea is actually to be agnostic to the type of data and pull in all the data from the clinical trials. So 4 pharma companies have decided that they're going to put all their clinical trial data into us. Riken in Japan has said we're going to throw in all of our data. And what's really the cool part for me is citizens. The patients in the arthritis internet registry have been getting online, and they're lined up and they say, I'll give you my DNA and I will tell you what happened to me, and I want my data as part of this process. So part of this has the arthritis internet registry coming directly in from patients, not from centers at all. They're just going online, putting their data in. And the winner for this, so we have 2 large journals wanting us to put it in there, but what I like, the main prize is the winner will have as their prize a human clinical trial automatically started with that classifier. So it will immediately go in, and so what does the winner get? An automatic clinical trial where citizens will get to enroll, we'll see whether what we found is right. And so we've bypassed the entire usual complex of who does what and times to get grants and things like that. That's sort of what it looks like. So I'm going to end here. The power of engaging challenges, think of it as allowing core experts that aren't where you are. You don't have to hire them, okay? Able to work on it. And it allows a wide range of incentives. And I will tell you, some of those incentives have included publication. I have a group we're working with where the incentive is automatic admission. We're working on a place where you could get tenure. Okay? We're working on the fact, I'm serious. We're working on a place where they're saying you get an automatic interview. So if you're wanting to become a faculty at a very prominent university, you will get an automatic interview as a faculty candidate because you've won the challenge. And if we can move so that the incentive structures that are coupled to papers, which is a recognition from your peers, the ability to have journal articles, et cetera, I think we'll get there. And I'm not going to talk about, unless a question comes, this amazing project we're doing with DARPA on melanoma, because I don't have time. So embrace the complexity was the first part of the talk. Remember, we haven't yet uncovered the rules. Begin to shift to doing data-driven science, build incentives to match that world, and this will not be affordable unless we figure out a way to engage the patients and get them involved. Thanks very much.